Welcome to foodfeedinfo.com. In this video, we will discuss briefly about regulation of fatty acid oxidation. Question is why regulation of fatty acid oxidation is required or it is so important, though all other chemical mechanism in the body, they are also being regulated. So if we look at the initial steps of fatty acid oxidation, particularly the beta oxidation, this oxidation consumes energy during the initial steps. So that's why those chemical reactions which are utilizing the body energy, they are more important. So they should be properly regulated to avoid the wastage of energy from the body. So it means it should occur in a way when there is a requirement of energy rather than it should be a continuous process. So that's why regulation is more important when the body fuel is at stake. So just look at the utilization or oxidation of fatty acids. So in the liver, there is a formation of fatty acyl CoA. So this formation is in the cytosol and it has two major pathways for its further utilization. The one which is its oxidation by enzymes in the mitochondria. So from cytosol, fatty acyl CoA is then shifted or transported to the mitochondria where it undergo beta oxidation process. And the second utilization of fatty acyl CoA is through its conversion into triacylglycerol and phospholipids by enzymes present in the cytosol. So the pathway taken by this fatty acyl CoA that depends on the rate of transfer of long chain fatty acyl CoA into mitochondria. So for the first pathway where fatty acyl CoA is transported into mitochondria, it is a three-step process which is carried through carnitine shuttle. So through carnitine shuttle, fatty acyl groups, they are carried from cytosolic fatty acyl CoA into mitochondrial matrix. So this is the rate limiting for fatty acid oxidation, and this is an important point of regulation. So when fatty acyl CoA get entrance into the mitochondria, then it undergo beta oxidation, and we can say the acetyl CoA is then oxidized once it is in the mitochondria. So if it could not get entrance into the mitochondria, it may follow some other pathway. So that's why it's a rate limiting step. So next, if we look at the biosynthesis of long chain fatty acid from acetyl CoA, which is in the cytosol, there is a production of malonyl CoA. So malonyl CoA is the first intermediate in the cytosolic biosynthesis of long chain fatty acid from acetyl CoA. So its concentration increases when animal is well supplied with carbohydrate. As carbohydrates, they are utilized for the production of glucose. So when there is excess glucose and that glucose cannot oxidize or stored as glycogen, then what happens, this glucose is converted in the cytosol into fatty acid for storage as triacylglycerol. So this malonyl CoA plays an important role because the inhibition of carnitine acyl transferase 1 CAT1 by malonyl CoA ensures that the oxidation of fatty acid is inhibited whenever the liver is an ample supply of glucose as a fuel and is actively making triglycerol from the excess glucose. Further regulation is through two other enzymes which are being regulated by the metabolites. So those metabolites, they signal sufficiency of energy. The first one is when NADH-NAD ratio is high, hydroxyacyl CoA dehydrogenase is inhibited. Likewise, the high concentration of acetyl CoA inhibit the thiolase activity. So you can see from the figure that these two are the major part of this beta oxidation pathway. So in this way, we can say the fatty acid oxidation is tightly regulated to ensure energy supply when it is required and to avoid the loss of energy through fatty acid oxidation when it is not required. Thank you.